So that was a really interesting conversation and a founder story like we have never really heard before on the show.
Yeah, Dennis really has an interesting background. So, came from a strong drug discovery background. He co-founded a company called Pimera Therapeutics, which is 10 years in and was partially bootstrapped at the beginning. Then they found funding. They asked friends and family. They really made this work despite a really challenging funding climate.
Yeah, definitely take a listen if you're interested in the various ways that people secure funding, how they get their company going when they are bootstrapping, and just a lot of really cool insights into all sorts of RNA topics and sort of where he sees the industry going. It's a great episode.
Yeah, absolutely. I think another really interesting thing that I learned is maybe leveraging other countries for clinical trials as a strategy. So take a listen. It's really interesting. And if you are on the brink of founding a biotech and you're not quite sure where to get funding, this one's for you. All right, Dennis, thank you so much for joining us today. We always start off with the same question, which is, what did you want to be when you were 7? What are you now? And how did you get there?
Well, it's nice to be on the podcast. Karina and Allison, thank you so much for your invitation. Appreciate that. It's funny, I don't remember whom I wanted to be when I was 7, but there are 3 jobs which I aspired to grow up to hold when I was younger before I ended up on my sort of current situation. I was quite an avid book reader when I was a kid, and I read all these wonderful stories based around archaeology and paleontology. So that was when I was probably around 11, 12 years old, these were my top choices. It's digging in the ground somewhere. I still sort of carry that hobby, which got kind of enhanced when my mom and I took a around 100-mile hiking trip through the Ural Mountains, which it's like a preserve, but instead of animals, it's a neurological preserve. So you can find some very interesting specimens there, like garnets just laying on the road in front of you. And it's also a very interesting geographical spot because Urals, they separate the Europe and Asia. And there's a place there, you go on this mountain road, there is a forest on the one end, the mountains on the other end, and there's a line which says Europe, Asia. And you can stand one leg in one direction and one in another. And that kind of prompted me to really wanted to grow up a geologist. But in the end, as we grow up, my interests change and some other priorities in life set in. Besides thinking about a career, I just decided to take the easiest route out. Both my parents are were chemists, unfortunately, but chemistry was something which was very easy for me as a kid. I got kicked out of the last year of high school from chemistry because of my unwanted comments to the teacher. So I went to Moscow State, and in Moscow, the higher education is a bit different. You select your specialty up front, and school is just like elementary, middle, high, college, graduate school is 6 days a week, and you take everything basically. I had to start the dozen types of chemistries, but on top of that, philosophy, sociology, economy, et cetera, which I found to be almost boring, but still you have to educate yourself. But when I graduated, I got a master's in chemistry. My work was recognized, and I was invited to come to States full scholarship for graduate school, UMass Amherst. The only catch was that I had to switch to molecular biology as my specialty, which was a little bit challenging because first discovery I made when I came to States, I did not understand half of what people are saying, despite learning English since I was 8. And second is that when I first came to the class, out of the half I understand from the cell biology course, I understand only 20% because I never took cell biology before in my life. It is the graduate course. But so it was kind of an evolution. But in a big scheme of events, I kind of stuck with the RNA as a sort of topic of interest, which I started investigating in 91, first as a chemist, then as a biologist, and then I did postdoctoral training at ISIS Pharmaceuticals, which is the number one antisense company, RNA-targeting company, been around since 89. And there I did a postdoctoral training for pharmacology and toxicology of RNA. And that kind of sealed my fate in a way. I did a lot of different roles within the drug discovery, development, translational research path, both from chemical, biological, and toxicological perspective. And I worked on other programs outside RNA, but it was kind of a unifying theme for me in a way, and led to the creation the company which I co-founded, Pimera Therapeutics.
That is amazing. And that trip you took with your mom sounds so beautiful and just incredible. What an amazing experience to get to go and do that. Can you tell us a little bit about, I want to make sure I'm saying it right. Is it Pimera?
Number 5, Pimera. Yeah, it has, even its name itself has an interesting sort of roots. We founded in 2013, and the reason for that was that the company I worked in the past, Silene Pharmaceuticals, was sold. And at the time, we had 3 programs. So one was in POL1 transcription, one was the CK2 kinase, and both of those were sold. But we also had a PIM kinase program, which kind of landed abandoned. And my partner, Dr. Mustafa Hadash, who was a head of medicinal chemistry at Silene, he wanted to do a PIM inhibitor. And that hence he came up with the name Pimera. And I said, well, we're not going to do that. We're going to do POL1. And because, as I said, I just found this RNA much more interesting to work with. And this is technology which I personally, with his help, discovered and co-developed with Professor Hannon, who is currently a vice dean at Australian National University in Canberra. And that was my passion. And I strongly believe that there is a sort of positive impact we can produce working in this front. So we kept the name from the original idea, but the company was refocused on the POL1 transcription and inhibition of ribosomal RNA.
Very cool.
Yeah. And so what's the vision that sort of drives this company? What is your mission?
Well, so a lot of discoveries in drug development discovery process are quite serendipitous. I'm sure you've heard the story about Viagra, right? That it's blood pressure. Then there is a thalidomide, which was originally for morning sickness, which caused some horrible birth defects. But in the end, started a new revolution in imides, lenalidomide, pomalidomide, et cetera, which are helping hundreds of thousands of patients with myeloma. So in our case, sort of this POL1 idea was born out of a serendipitous discovery that when I was hired by Silene Pharmaceuticals, they said they had this compound, CH3553 corploxin, which was originally designed to target the DNA structure within the promoter of MYC oncogen and suppress its transcription. And they were ready to go in the clinic. Everything was peachy. The only problem that did not inhibit MYC. So I was hired to find out what does it do. And I serendipitously observed drug fluorescence that accumulates in nucleoli of cells. And that's the place for ribosome biogenesis. That's where place the ribosome DNA synthesis occurs. So I had an idea, maybe that's how it works. And it did. And that gave us an idea of just starting a program looking at the POL1 inhibitors independent of their mechanism. And the first true generation, because corploxin had some general effects as well. POL1 inhibitor was born in our like 6461, which was sold to Sanofi Biosciences and being currently developed in several clinical trials for oncology. And it was as a first generation drug, it had its own liabilities. I mean, it was an IV, it had a very long half-life. There's some other issues which we wanted to address. And with Pimera, we knew exactly what we needed to do in terms of what needs to be improved and et cetera. And we basically found Pimera to address any potential liabilities on that and come up with some other new ideas. And basically, we were able to raise some money, not a lot, but because we were lean, mean machine, so to speak, relied heavily on consultants, CROs, wrote a lot of IOUs in the beginning. And that's kind of one of the advices I can give to people who are starting their own company. Find people who will trust you enough to give you credit, because in the beginning, there are certain things which you wouldn't even think about, such as lawyer fees, for example, are right up front for all your corporate needs, at least, that require significant spending. And in our case, our first raise was basically the 3 of us. There's another gentleman who was, but unfortunately, he left the company pretty early. We just put for $10,000 each. And that was our seed round, $30,000, which for some businesses is a lot of money, but not fun trying to discover and develop.
Wow. So this was all getting started. Pimera was getting started in June of 2013. I think, is that about the right timeline?
Yes, we were technically founded in April, but all the paperwork were filed with the state of Delaware in June. So that's the official birthday.
So I definitely want to touch on this leveraging consultants and who you bring at the early stages. But just to set the landscape now, it's been 10 years. What does the company look like now? How many employees do you have? Are you still leveraging some fractional talent? Can you take us through what that sort of looks like?
Absolutely. So the company never had more than 3 full-time employees. And that was kind of our strength in a way, because second being the fact that Janssen, what used to be Janssen Labs and now J Labs, were also very kind to us and kept us for many years beyond the original expectations. And right now, company has 2 people full-time. We are in the phase 1 right now in oncology, and we extensively leveraged our contacts with both academic and medical community in Australia, and currently is running clinical trial at their number 1 cancer center, their McCown Cancer Center in Melbourne. And there is another benefit doing a clinical development in Australia is that the government there gives you a significant amount of rebate on every dollar you spend. So despite Pimera being a small operation, technically we have 3 offices, San Diego, Houston, and Melbourne. So there is a Pimera Australia.
So I find this so fascinating because we hear a lot. We work with in companies in sort of a venture environment a lot. And so it's very much the traditional fundraise. They go for their seed, their ABC. You've just done things a little differently. And that is so fascinating. So can you tell us a little bit about the alternative? So the one thing you mentioned already is doing your clinical trials in Australia. That is such an interesting perspective. We actually have not heard anybody talking about other countries for clinical trials on the show. What other things can you say, if you need to be lean and mean and scrappy, how else have you done that?
It's good to have on your team a person who knows how to market. And that's my partner. He was born with that talent. He allowed us to really adhere to our budget, which was always pretty slick. But the thing there is, one of the main routes to success in drug discovery is you need to eliminate the bad things first. And we knew pretty well what's going to play, because we had a significant amount. We had 9 months to think before the physical wet work, so to speak, began. To plan what exactly it will be, the screening cascade, what the molecules we're going to look at, what kind of indications we'll be thinking about, bring on board collaborators with whom we worked in the past. And that's another, I think, one of the most crucially contributing factors was our collaboration with Professor Hannon, who was started at the Pimera Cancer Center and now is at University of Australia, National Australian University. His lab also specialized in having interest in Botswana and Rapson biogenesis, had an access to an amazing amount of models, which otherwise we would not be able to secure. Even nowadays, despite the successes of all the CRISPRs in the world, there's still a limited number of transgenic mice, which can be a good representative model of the disease, in particular for oncology, because typically oncology is a multigenic disease, not monogenic, some genetic disorder. So having him personally interested in potentially help the patient, but also for advancement of science was a very beneficial sort of symbiotic arrangement where allowed us to succeed.
Yeah, so academic collaboration, certainly a big one. We definitely see most founders trying to do a lot of the science in-house. So that's a really neat strategy to work with collaborators who have maybe some funding, some graduate students. They're working through things that are, in parallel.
Well, exactly. I mean, even if you do find transgenic models, and as I said, there are some, the study will cost you, it's around $250,000. So one study. So that's something which can really add up eventually on your budget.
Yeah. Wow. So budgetary constraints aside, when you work with someone who's maybe not an academic partner, but a CRO or a consultant, what's really important to you when you're vetting different companies to work with and partner with?
So I think there was developed sort of as a rule of thumb agreement that we will only employ CROs for things which do not require tinkering. If it's a pretty straightforward, like toxicology study, just follow the protocol, collect all these tissues, run all these analyses, provide us the data, we will look at it. All right. But for something which is more innovative or more requires trying different things, that's unfortunately CROs are usually not good at. But again, we capitalized heavily on the globalization, worked a lot with China, there are some very good vendors there you can find, worked for chemistry as well as the pharmacology, toxicology, pharmacokinetic studies, which were done not half, most in China, but we're also working with Australia because they're also working through some, and that's another thing. A lot of bigger university has so-called core groups with whom you can also collaborate at much more reasonable prices than with the CROs. But again, sometimes they'll ask you, like, we used to collaborate with core here at San Diego, not at Silene, and they're kind of have to go there yourself. But there are instances where they will actually do the whole work for you.
As far as managing those CROs, do you have somebody on your team who specializes in that? Does everyone sort of do that ad hoc? And how much management is there?
Well, it really depends on the study. I mean, my partner and I would kind of split that. He's was all the chemistry side, mine was all the rest. Oncology, toxicology, PK, et cetera, et cetera, et cetera. I think that also having, for example, for toxicology, we were also blessed to find really good expert consultant who is also a San Diego native, Dr. Grace Berman. And she was, because in any CROs, there are employees who are better and there are employees who are worse. Not naming any names in particular, but having somebody on your team who already had experience working with a particular CRO, or talking to your, not necessarily even having on the team, but knowing somebody who can give you advice in terms of, for example, what study director should you ask for? Is there any potential pitfall which you should look be upfront that this CRO is known for? This can be very helpful.
I have to ask on a totally different note. Could you tell us a little bit about like what your day-to-day is like? I mean, you're managing communication and contracts and talking to people all over the world. And I imagine you either work 24/7 or you have really figured out a great way to manage all of the different pieces.
I'm still human and I need to sleep right now because I left as a full-time employee of Pimera. I'm not involved there right now as much, but when I still was there, the biggest challenge was in a way that we worked across the globe. So sometimes, the meetings have to be taken not in the middle of the night, but later in the evening or early in the morning. So 7 a.m. was not completely unheard of for sometimes 6 a.m. So, and that's just part of the deal. But again, in our case, there you have to sort of always maintain contact with those CROs and actually your collaborators as well, because that's one thing. The academics can get a little bit sometimes drip off because something which is maybe very interesting from a scientific perspective does not necessarily add anything to the drug development. That's sort of where you need to start communicating with them and making sure that goals are aligned. But a lot of time is actually, it's just reading because there's so much an explosion of new information every day that staying on top of it does require reading quite a few hours a day, pretty much. Usually when I wake up, I'll open and start looking for what were announcements from the East Coast, for example, because I wake up at 6. That's already not there.
Do you have any AI tools that you use to help you gather information or help triage that process?
Well, my brain.
Your brain?
Not A, but I at least.
You have eye tools.
I love that.
Yeah, I'm exploring different tools the information has always been a challenge. There's so much to consume to stay on top.
I mean, in my mind, between PubMed, Google, and Google Patents, you can find all information you need. The issue is you need to know what question to ask. All of them, the information is out there. That's the challenging part, is know what you're looking for. And works for me so far. I'm not going to knock on AI, and I'm not going to say never. I mean, and for a lot of things, especially right now in the field of RNA, for target discovery, for example, or looking at the different regulatory elements throughout the sequences, or establishing the connection for signal nucleotide polymorphism to potential pathology, it cannot be done by I without A. So that's where this comes very handy, and that's what we use in the work often. But for searches, still do the old-fashioned way.
What excites you? What are you seeing in the news right now or coming through that you're like, oh, wow, that's super cool, and I can't wait to see this get developed?
All information you need. The issue is you need to know what question to ask. The part is know what. I'm not going to knock on AI, and I'm not going to say never. I mean, and for a lot of things, especially right now in the field of RNA, for target discovery, for example, or looking at the different establishing the connection for into potential pathology, it cannot be done by I without A. So that's where this comes very handy, and that's what we use in the work often. But for searches, still do the old-fashioned way.
to stay on top?
Google 1989, but there was no enthusiasm because I think that there was no recognition of the potential. The antibodies were stealing everything with it. Obviously, great result from the oncology, et cetera. But RNA was sort of this orphan child, which was usually neglected until 2020, pretty much. And at this stage, although there were other companies on this, on Island, et cetera. But since then, there has been an explosion of different modalities. And I think that there's three main paths that nucleotide-based therapeutics are currently advancing in. And one is discovery of different modalities. It started with antisense, and it's RNA, then mRNA, and now there is, God knows what else, there's circular RNA, self-replicating circular RNAs. There is non-coding RNA-based therapeutics. There's Alterna, which they're trying to make a tRNA for nonsense suppression, and et cetera, et cetera, et cetera. And that's very exciting that the amount of sort of possibilities is opening up. Second, and that's where the AI sort of comes in, is the use of computational methods which allow you to analyze the data which is being produced. Because by now there's already so much genomics and RNA-omics data which just sits there from human sequencing, which needs to be properly analyzed to be understood of how certain changes, genetic, epigenetic, et cetera, are connected to pathology, and which of them are causative, which are correlative, and et cetera, et cetera, et cetera. And that's where computers come really, really handy. And the third, it's a little bit on the slower pace, but there's some interesting developments there, is its chemistry. It's a chemistry which improves your drugs, especially in the biggest challenge of oligonucleotide therapeutics, pretty much, which is delivery. Right now, the liver is kind of covered after introduction of GalNAc, the CNS and eye, because you can inject directly in those spaces, maybe skin to a degree, but this is definitely not everything, right? I mean, there's new work going on in muscle, in lung, and in kidneys. Regulus, for example, the company I work for, has some very interesting technology for the kidney cystic disease. And I think that we live in a wonderful time for that, for the expansion of the field.
Yeah, I'm seeing a lot of computational chemistry roles being posted suddenly, which I feel like, so I'm an RNA biochemist by training. Moderna was my first client, and this was back when they were working on that RNA vaccine idea, and everyone was started to talk about how they were maybe a unicorn that wasn't actually going to do anything but make vaccines. And there was like that was a derogatory thing. And then, COVID hit and they were like.
But I felt that there was a serious interest in oncology in the beginning as well.
Oh, yeah, definitely. But the thing that they were able to gain traction on and monetize, or that looked like it was going to be monetized, was the RNA vaccine unit.
Well, $100 billion from United States government did not hurt, I assume.
No, I'm sure it did not. But I just remember that sort of souring around that RNA field.
Yes, and it's specifically in oncology, simply because the delivery could not be achieved. When I was at ISIS, now Ionis, they had a leading program compound called Therapeutic, which is a protein kinase C inhibitor, and they put it in lung cancer trial, and it failed 2 phase 3s, which was heavy toll on the company. And one of the potential reasons in my mind is that they simply wouldn't go to the tumors in the sort of needed concentration. And there are a lot of new strategies which are currently being developed for specifically tumor targeting. I'm really looking forward to see what can be achieved.
Yeah, same. That's really exciting. So what have you loved and what have you hated about being a founder? Give us the top 5% and the bottom 5%.
I think that when you start the company, if you're not in the position where, you can retire already, all right, it's going to hurt. But there's a significant financial component where, you're missing, like, for example, for first 18 months of Pimera, it was me and my business partner who put money in it, not vice versa. Only after we got our initial, not even first or second initial investment, were we able to pay a just token salary for ourselves. But the most exciting thing is where you understand that you can do it, that the idea is working and that you do know enough, that you have people working with who can help you enough to make this idea into reality and to go into the clinic and hopefully help patients in need.
The mission. Yeah, I think that mission is what we hear a lot, and the hope that we can help humanity and see that come to fruition. I love that.
Well, I mean, we can talk about, like, for example, potential pitfalls about taking new technology from academia and trying to develop it. Like in our case, we have, and this is sort of, I feel it's my strength. Some people may say not, but I have never worked in academia. My first job was already in the industry. I've met a lot of people who switched, and one of the biggest challenges which they face is they get attached. And that's so true for academia, because this is your project, right? You're working it 5 years, and in the year 3, something stops working. You're not abandoning it because you already dedicated 3 years to it. In industry, it's completely different. It doesn't matter how much time you dedicate to something. If you see that it's not working, you need to immediately abandon it, because that's sort of throwing good money up to bad. There's a lot of instances, and a lot of instances led to downfall of company where they were not nimble enough to switch to something which works. And that's why, like in the beginning, we did not completely discard, for example, PIM idea. It was, if the POM1 is not taking off, or cannot make inhibitors with a completely new chemistry, which we came up with at the time when we were thinking about the company, then we will switch to something else. But we were lucky that compound number 12, compound number 12, which we made out of all our library, in the end turned out, Pimera 116, in the end turned out to be the lead, which is currently, in a clinical trial.
That's really interesting. I was speaking to someone the other day about, it's a little bit of the elephant in the room, right? Like, how far do you take something before you just say, like, this isn't going to work? And, I'm curious, did you have like a set time frame to where you were like, if our initial idea doesn't work, we pivot? And what was that discussion like? Because we do talk to a lot of founders, and the question always becomes, how long do you chase the dream before you think it's either a pivot or it's just not going to happen right now?
Well, in our case, we also had a benefit of the first gen being already in the clinic and clinical data being released publicly while we're doing all that. And there were some promising results there. And we had access to exactly the same models we tested the first gen drug on, continuing collaboration in Australia. So we had a pretty good idea that we can do as well as the first gen or better, right? And our drug is oral, which is a benefit instead of IV injections. And if we can't, we can't. So the progress continued. We saw activity pretty early in the models, which was superior to the first gen. We saw that tolerability is clearly much better, and that gave us confidence. And there, I'm not saying that there are problems which at the first sign of trouble, you should abandon ship. No, but like, for example, if you're going into already clinical trial and you have problems scaling up, I wouldn't abandon the program either. I'll start working on how to scale up, right? But if your science or idea does not work in the beginning or even in the middle, like, for example, sometimes you can pivot. It happened to me in my career once. When we discovered about the POL1, and when we switched the company, Selling Pharmaceuticals, whose CEO, Bill Rice, he was very receptive to the idea and actually helped me make it happen. And we switched to 2 different programs looking for POL1 inhibitors. And we chosen very different paths. One was a shotgun, where you don't care what your molecular target is. You have a functional screen and you put a library of compounds through it, and then you take hits from that and go from there. And another approach was to select a target from the literature and then work towards molecules inhibiting that target and then connect it to your pharmacological response. So I was in charge of discovery of the first program, and my colleague, Dr. J. Adam, was in charge of the second. And when in the end, the 2 compounds came out, so 5461 and 4945, 5461, we had no idea how it worked in the beginning, but we knew that it's a selective and potent inhibitor of POL1. And with 4945, we knew that it's a very selective inhibitor of CK2, and it did it pretty well. It had no impact on POL1 transcription. So at this stage, we had already a molecule which was a lot of invested in, but which did not work through the proposed mechanism. And instead of dropping it again, we pivoted. We started putting in charge of translational research project, which start looking at other potential applications of this drug. I'm not saying you have to throw everything away. It's just you have to be very careful, not try to make CK2 inhibitors inhibit POL1, because apparently that's not going to happen. All right. But see what else can you do. And we were lucky working with Adam. We discovered several potential combinations, like CK2, for example, regulates DNA damage repair. And that's what is in the clinic right now in combination with 2 DNA damaging agents. So phase 2. It's just, you have to be ready to follow the data. Disperse of your original notions if they're not working out.
Yeah, you have to be adaptable.
Yes, adapt to that.
You strike me as somebody who's very business minded. And I think that probably is a result of not, like you said, not having an academic career or a lengthy academic career, both business minded and also very tenacious. So to have followed something through on a shoestring budget for as long as you did to push it into the clinic, that's a lot of tenacity. It's a lot. Can you tell the other founders that might be listening, kind of a little bit about that journey and what did keep you going when things got a little bit tough?
Well, I think that it's very important to understand that starting your own venture is an investment. You find people to give you money, and that's just the money, but you are investing your time, years, primarily. I stayed full time with Pimera for 7 years until we went to the clinic, basically. And just be ready for that. Don't look at the time sort of where you could have been making money. Yes, there's hopefully payout down the road, and it'll be much more sweeter because you will know that all of it is your work. And just chase your dreams, I guess.
Yeah, it is a long process to bring a drug all the way through.
It is. I mean, we had some unfortunate snafu, which delayed us just a couple of years, nothing to do with us, but 5 years of work basically was put out from the idea into the clinic. And that is on the sort of considered to be on the shorter side, usually, because it can take up to 10 years sometimes. Although nowadays, a lot of new programs, I did a bit of consulting. I talk to everybody wants to be in the clinic in 3 years. So that's the new rule. We'll see how it will work. I mean, and for some therapeutics, it is obviously easier, especially for oligos, because chemistry there. I mean, if you know your sequence, you know what your sequence of your compound is going to be. When you don't have any chemical start, some small molecule problem, that can take a lot of time.
So what are you doing now? You said that you're not with Pimera full time. So are you doing some consulting? Are you kind of just taking a breather?
Well, in a way, I do a bit of consulting, not a lot, but I'm just thinking about what's new.
What do you want to be doing?
I don't want to abandon my passion just yet. I want to stay in the RNA field. There's a lot of, as I mentioned, there are a lot of new interesting modalities coming around. So I'm sort of looking what would be the best place to apply myself to.
Yeah. If you're talking about doing some consulting, what is the consulting model that you're thinking about and what types of companies are you working with?
So there are different opportunities. I have CDA funds. Unfortunately, I can't talk to the names. But there are some actually I recommend. There are certain CROs who what they do is they connect clients to the specialists. And working with them can be quite beneficial. They serve as a bridge between the need and the provider. It's not a full-time gig, but they're sometimes where they need a specific specialist advice. And that's where I can come handy.
That's really good advice, though, to share out with people who maybe don't know that that's an avenue that they could look at and connect through. Maybe there's some opportunity there for other people to weigh in and offer some expertise.
Of course. I mean, this is more, and that's another benefit from COVID. The globalization and remote work, so to speak, hybrid work, has become a norm. Right. So it just makes more sense if you have a question to ask somebody whom you don't need 23 other hours out of the day.
Yeah, I sort of think about at the beginning, we were talking about your model where you have folks spread out all over the world. You are working with CROs in China. You're doing your clinical trials in Australia. That is still a little bit novel, but I bet if somebody were to listen to this podcast a few years from now, they would think, well, of course, everybody's doing that. That's just normal. So I do think we're moving in that direction because I hear more and more people, of course, the CROs in China, we have been using, a lot of CROs in China for years, but clinical trials or teams in other countries that are doing meaningful research, that is somewhat new.
It is in a way, but then again, this is sort of depending what your goal is. For oncology, in a way that the beauty of it, or rather the horror of it, is that there are patients everywhere. This is something which is hours out of the day.
How does that affect the approval process or patents in the US?
I am so glad you asked that because I was just thinking, as the non-scientist, I was like, I'm going to take one for all
It
of the people
is
out
in a
there who
way,
are
but then
operations people and
again,
who have no
this
idea
is
how this
sort
would
of
work.
depending
I
what
figured
your
that was
goal is
standard
for
knowledge. So
oncology
yes,
in a way
please
that
tell us
the
that. I'm
beauty of
so
it,
curious.
or rather the power of it, is that there are patients everywhere. This is something which is hours out of the day. It is in a way, but then again, this is sort of depending what your goal is for oncology in a way that the beauty of it, or rather the power of it, is that there are patients everywhere. This is something which is hours out of the day.
Now, did you know that when you were, kind of starting to work
It
with
is in a
Australia, or was
way,
it one of
but
those things
then
where the
again,
opportunity
this is sort of
presented itself
depending
because of the
what your
network? So
goal is
then you were
for
like, oh, great, look at
oncology
all
in
this
a
other
way
great
that
stuff that
the
also
beauty of
kind of
it, or
correlates
rather the
and will
power of it,
actually
is that
transfer
there are
into the
patients
US. Like,
everywhere.
did you kind of
This is
build that
something which
knowledge
is
out as
hours
you were
out of the
deep
day.
in the process, or was Australia a target prior knowing that?
It is in a way, but then again, this is sort of depending what your goal is for oncology in a way that the beauty of it, or rather the power of it, is that there are
Yeah,
patients
we learn new
everywhere.
things
This
every
is
day
something
by
which is
talking to
hours out
people
of the
and
day.
by going down these paths. But I imagine that when you're actually in the thick of it, sometimes you have to kind of learn what other countries do, what other clinical trials scaffolds there are. Tell me about your thoughts around a company at this stage, because I see this a lot where it's phase 1 trials, and then the hope is to license it, to your point, to maybe a pharma company to finish the development. What are your thoughts around that?
It is in a way, but then again, this is sort of depending what your goal is for oncology in a way that the beauty of it, or rather the power of it, is that there are patients everywhere. This is something which is hours out of the day. It is in a way, but then again, this is sort of depending what your goal is for oncology in a way that the beauty of it, or rather the power of it, is that there are patients everywhere. This is something which is hours out of the day.
Yeah, that's fascinating. So, is that the end goal, do you think, to license this? Or do you want to see this all the way through?
It is in a way, but then again, this is sort of depending what your goal is. For oncology, in a way that the beauty of it, or rather the horror of it, is that there are patients everywhere. This is something which is hours out of the day.
Yeah, definitely not. Some of our clients, increasingly, there's been a model, and I actually think, I wonder if we're going to see this change. But for a while, there was a model where you mentioned the sort of 3-year target to clinic. That very much is the plan for many companies that are spinning out of some of the larger VCs. And they're putting into place in year one, they're putting into place the folks that are going to be on those, on that clinical, the regulatory side, because it takes a long time to spin that up. It's not like, okay,
It
we're ready
is
to go
in a
to
way,
trials,
but
just hire
then
the people.
again,
So
this
much
is
has to be
sort of
done. And
depending what
the
your goal
burn,
is. For
the cash
oncology,
burn
in a
for that
way
is
that
incredible.
the beauty of it, or rather the horror of it, is that there are patients everywhere. This is something which is hours out of the day.
Yeah, that's the other missing piece of that, is that these companies, they do have a strong lead when they're spun out of these VCs. It's typically that they've partnered deeply with an academic who's come in
It
to be
is
their
in a
founder.
way,
So
but
yeah,
then
it's
again,
definitely
this is
it's
sort
a
of
different
depending
model and
what
it's a
your
different
goal is. For
mindset. And we
oncology,
see
in a
the
way
gamut where
that
we
the
have
beauty of
smaller
it,
and leaner
or rather the
biotechs like
horror of
yours
it,
that are
is
very
that there are
much
patients everywhere.
bootstrapping this
This is
to some
something which is
extent,
hours
getting
out of the day.
funding, at the right stages, and then hoping that the outcome is a licensure, because that really is a nice outcome.
It is in a way, but then again, this is sort of depending what your goal is. For oncology, in a way that the beauty of it, or rather the horror of it, is that there are patients everywhere. This is something which is hours out of the day. It is in a way, but then again, this is sort of depending what your goal is. For oncology, in a way that the beauty of it, or rather the horror of it, is that there are patients everywhere. This is something which is hours out of the day.
Yeah, no, that sounds great. JLab sounds really interesting.
It is in a way, but then again, this is sort of depending
No,
what your
that's
goal is.
really
For
good advice.
oncology, in a
I am
way
so
that
interested
the
in your
beauty of
answer
it,
to
or
my favorite
rather the
question,
horror of
which
it, is that there are
what
patients
is
everywhere.
your
This
favorite
is
book
something
that
which is
you think
hours out
everybody
of the
should
day.
read? Oh, great. We'll take all 13 recommendations.
It is in a
Oh
way,
yeah, I've
but
read
then
that.
again, this is sort of depending what your goal is for oncology in a way that the beauty of it, or rather the horror of it, is that there are patients everywhere. This is something which is hours out of the
Yeah, I
day.
agree with that. It's really funny. I'm also a big fantasy person, and my husband and I have both read all of the books of Wheel of Time, and so we were really excited when the show came out, right? Of course, and it's very different and true to TV. They made it very racy, which it is not at all in the books. So they've taken some liberties, we'll say.
Well, I've never read it, and so it's going on my reading list. So thank you both. Sounds like a great read.
And so we are going to link your LinkedIn into our show notes, but where else can folks get in touch with you if they want to learn more?
It is in a way, but then again, this is sort of depending what your goal is for oncology in a way that the beauty of it, or rather the horror of it, is that there are patients everywhere. This is something which is hours out of the day.
Yeah, amazing. Well, we'll send people over there if they want to get in touch. Thank you so much. This was really informative. Yeah.
Yeah, Des, it was a pleasure chatting with you.
Thank you so much, ladies. It's such a pleasure to chat with you again, Alison. Karina.